Hello everyone! This week in lab we're going to talk about isolation methods, isolation of bacteria. In this PowerPoint we're going to focus on streak plates, and this is an image right here of a streak plate. A streak plate technique is basically used when we want to isolate a pure culture of bacteria. So let's say you have a mixed culture, basically a mixed different species of bacteria, and you want to go ahead and achieve a pure culture. We usually do a streak plate, and it will end up looking something like this. And we get those single colonies right here. And we're going to talk about how a streak plate can help us achieve a pure culture and getting a pure culture of that organism. We're going to discuss the details of how we usually culture bacteria and how we achieve that streak plate look right here. Let's first talk about a few vocab terms that you need to be familiar with. And the first one is the term culture, which we've used already before, but just to refresh your memory, a culture is basically growth of microorganisms. So anytime we talk about growing bacteria, instead of using the term grow, we use the term culture. So when I say I'm going to culture my bacteria, meaning I'm going to grow my bacteria. So you can use it in a verb format, or you can use it as a noun. I have a culture of bacteria, so meaning you already have growth on your plate or in your broth. So that's why we say in or on your media, because you can have cultured bacteria on a plate, on a slant, in a broth. So anytime you have growth of bacteria on your media, we refer to that as a culture. Now, if you take a look at this plate right here, this agar plate right there, you can see those colored circles right here. Now, what do we call those masses that you are seeing on the plate? We refer to them as colonies. So colonies are basically those visible mass of bacterial growth on your plate. And you can actually take a look at those colonies and you can determine whether you are looking at a mixed culture or a pure culture. And I'll explain that in a minute. But as you can see, those are the colonies right here on your plate. And what's a colony? A colony is basically a mass of bacteria, and they are basically consisted of many clones of one bacteria cell. So one bacteria cell keeps reproducing and replicating itself, and you get this visible mass on your plate, and we refer to that visible mass as a colony. Now, looking at these colonies, right here on this plate, you can see that not all your colonies look the same. So you can see that we have some colonies that, for example, are yellow in color, orange, white. And so one thing you notice is the variation of color that you're seeing on this plate. Another thing you can notice is the variation of size of these colonies, their size. They're not all the same. So you have some colonies that are really tiny, some of them that are medium sized, some of them that are like much larger. So you can see variation in color, variation in size. You can also see variation in texture. So you might notice that, especially like if you zoom in on the plate, you might notice that some colonies, they look wet or shiny. Some of them look matte. So texture-wise, there could be a difference, and you can kind of see some differences right here. See how this one is like more shiny than this colony right there. Another difference that you can spot on this plate is even the margin of the colonies could look could be different. So one of them might have like very smooth edges, kind of like this one. Um, take a look at this one, for example, that white one right here. Notice how like its edges are not very smooth. So even the margins of the colonies right here on this plate, you can spot some differences in the margins of the colonies. And then even the colonies themselves, when you take a look at them, you can see some of them might be raised. Some of them might look pretty flat on the plate. Um, why should you spot all of these differences when you look at a plate? And you got to make sure that you are looking for all of these differences that we've just discussed, because that's going to give you an idea whether you are looking at a mixed culture or whether you are looking at a pure culture. A mixed culture is basically when you have multiple species of bacteria cultured on that one plate. And this plate right here, it is a mixed culture. How do I know it's mixed culture? Because of all of the differences that I've just laid out, right? The difference in color, the difference in size of the colonies, their margins, their textures, and then as I said, some colonies are elevated, some of them are like pretty flat. So see how this one kind of looks elevated, this one looks flatter. So all of these differences tell me that I'm looking at a mixed culture plate, meaning that I have multiple species of bacteria cultured on that one plate. Now, if you don't spot any differences and the colonies all look exactly the same, then you're probably looking at a pure culture, meaning that you're looking at a plate that only has one species of bacteria growing on it. So one of the isolation methods that we are going to concentrate mainly on this PowerPoint is a method called the streak plate, or you achieve something called the streak plate. And the streak plate technique is basically used for the isolation of pure culture organisms. So when you want to go ahead and isolate and get a pure culture of bacteria, we go ahead and we do the streak plate method. And again, we isolate those pure cultures in the form of single colonies. Okay, so when you get those single colonies, then you can achieve getting those pure cultures. And I'll explain how you can do that. And as we said, when you look at your colonies, you want to look at their color, texture, size, margins, because all of that would give you hints whether you're looking at one species versus variations of species of bacteria. And as I said, the streak plate method is going to be used to help us achieve and isolate pure cultures of bacteria. Now there's another plate that we're going to discuss more later on called the spread plate. And the spread plate is mostly used to count the bacteria colonies. So if we want to count how many colonies do I have, I go ahead and I do a spread plate. Now you might ask, well, why would you want to know how many bacteria colonies do you have? Because you might be asked, what is the concentration of your bacteria sample? Okay, so like about how many, bacteria cells do you have, let's say in a liquid broth medium. So you can't just look at a liquid broth medium and estimate, oh, I think I have 100,000 bacteria cells per mil. So we can't just guess or estimate. So we actually have to do some calculations so that we can figure out exactly how much bacteria do we have in this medium. And so one way of achieving that is we go ahead and we do what's known as the spread plate. And usually when we go ahead and we do the spread plate method, and I'm going to show you how we do it, we could start off, as I said, with a broth culture. So it's a liquid medium, and we usually dilute it out. through a process called serial dilution. And then after we dilute out our broth medium, which has been inoculated with our bacteria, after we go ahead and we dilute it out, we go ahead and we streak, or we go ahead and we inoculate our plates. So we go ahead and we inoculate our plates, and we go ahead and take a look at those spread plates that we've just inoculated, and we count the number of colonies, and based on the number of colonies we can actually tell the concentration of our original batch. And again, we're going to talk about the spread plates more in detail later. As I said, this PowerPoint, we're mainly going to talk about the streak plates and how they are used to isolate pure cultures. So as I mentioned before, a streak plate is going to be used when we want to produce isolated colonies of bacteria, let's say on an agar plate. And we want to go ahead and separate the cultures of bacteria from a mixed culture. You want to go ahead and purify and isolate a particular strain, okay, from multiple species that you might have on your plate. So to achieve that, to go ahead and isolate particular strains of bacteria from a mixed culture, we go ahead and we do the streak plate method. To kind of give you an example, let's say that you went ahead and you got a sample from your patient, okay? You picked up a sample, you know, let's say from their nose, or you went ahead and you swabbed and you took a sample from their throat, okay? Would you think that you would find only one pure culture in that sample? Absolutely not. You're going to find multiple species of bacteria from, you know, from a urine sample, from a sputum sample. You're definitely going to see multiple species of bacteria. So if you want to go ahead and you want to isolate, okay, and get pure cultures and isolate particular strains from that mixed culture that you have, you go ahead and you do the streak plate method. So how do we achieve that streak plate method? Let's go ahead and kind of take a look at the steps together, and then you guys also have the virtual lab to look at, and there are helpful videos for you to look at. It's going to help you out see the steps, okay? And you guys need to know the steps of the streak plate method and how do we achieve, that end look that we're going to get right here. So the very first step is to go ahead and pick up the tool that you're going to be using. And so the tool that we're going to use is the inoculating loop. So first thing you want to do with your inoculating loop is definitely, the very first step is I want to go ahead and sterilize it. So you sterilize your inoculating loop by passing it into the Bunsen burner, kind of like that. And in this image right here, they're not showing you that step, but this is the very first step, okay? So right here, okay, you should have a step that kind of looks something like this, where you grab your loop, you sterilize it by passing it into the Bunsen burner, and you pass it into the Bunsen burner until the loop turns red hot, okay? And then wait a few seconds to allow it to cool, and then go ahead, use it to pick up your bacteria sample, okay? Whether you're picking up that bacteria sample from a liquid medium or like a broth, or whether you're picking a sample of your bacteria from a plate, okay? By the way, if you're picking up a colony from a plate, you don't have to grab a huge chunk, okay, of bacteria. You could just pick up like again, just like tap your colony, grab some on your loop, and then what you're gonna do is you're gonna go ahead and you're going to streak you know, using your inoculating loop that now has bacteria on it, right? You're going to go ahead and you're going to streak very gently. You don't want to gouge your agar or anything, very gently over a quarter of your plate. So we kind of call this the first quadrant, okay? So about like a, you know, you're going to, about a fourth of your plate or so, okay? You're going to go ahead and streak. And do you see how you streak back and forth? So you keep streaking back and forth, without going back, by the way, into your original, into like the original sample. So you never go back into the original sample, okay? So you picked up your sample already, let's say from another plate, and then you go ahead and you streak back and forth like this, and make sure that you are not leaving a lot of space. Do you see how the lines, this is really important, see how the lines, again, you are not leaving any space as you're going back and forth. This is super important, okay? And then after you're done, okay, kind of going back and forth, and you've made sure that you've streaked that first quadrant really well, you go ahead and you flame your loop. So after you're done streaking that first quadrant, you go ahead and you flame your loop one more time until it turns red hot, and then wait just a few seconds. And then what should you do? You go ahead and you pick up, so you go right here on the edge of your first quadrant, okay? Kind of back to where you've already streaked, right, in quadrant 1, and you go ahead and you extend the streak downwards, okay? So it's kind of like you're grabbing, some of your bacteria that you just deposited right here, right? And you're dragging it down. And then you can go back into the first quadrant a couple of times, and that's it. Okay, and then stop going into the first quadrant, and you're going to go ahead and keep streaking back and forth into that second quadrant. So one more time, this is super important. Notice how when I'm streaking my second quadrant, I first have to grab some of that bacteria from the first quadrant, and then I go down. I can go back up, okay, but no more, you guys, than 2-3 times, and that's it. And then right here, notice how all of these lines, you are not to touch the first quadrant at all. Okay, and then what am I gonna do after I'm done streaking my second quadrant? So after you're done streaking your second quadrant, reflame your loop one more time. Okay, and the reason why we keep reflaming our loop is to prevent contamination. Okay, so reflame your loop until it's red hot, wait a few seconds, and then you're going to streak your third. Again, what am I going to do? I'm going to go to the edge of my second quadrant, pick a sample of the bacteria that I just deposited, and as I said, you can go back into the second quadrant a couple of times, and that's it. And then go ahead and streak your third quadrant. Okay, then reflame your loop. Do the same thing, go to the edge of the third quadrant. You can kind of, as I said, go back and forth into the third, and then go ahead and streak your fourth quadrant, okay? And reflame your loop, and you're done. Okay, so at the end of the day, your plate is going to look something like this. You have 4 quadrants, and what we've achieved, if you guys think about it, okay, we've basically diluted our sample. How did we dilute our bacteria sample? Right here, the first quadrant, this is where you're going to have the heaviest growth. This is where you're going to have heavy confluent growth, or we basically refer to it as a lawn of bacteria, right? This is where you're going to have the heavy, lawn of bacteria growth right here. Then the second quadrant is going to have less growth. Why? Because if you think about it, when we went and streaked the second quadrant, I didn't go back to my original sample, right? Never go back to your original sample. What we did was we went to the edge of quadrant 1, picked up some of the bacteria, right, that we just deposited on there, and we went ahead and we streaked our second quadrant. Then your third quadrant is even going to have less bacteria growth. Why? Because your second quadrant is already a bit diluted, and then you're going to go ahead and dilute it even further, okay, in your third quadrant. And then your fourth is even going to have less and less growth because, as I said, you're picking up some from the third quadrant, which is kind of like how I said, it's already diluted. So as you can see, your bacteria growth is going to keep decreasing as you're moving, through your quadrants. And that's going to be helpful because this is where you're going to see your single colonies in quadrant number 4. So in quadrant 1, you are not going to see colonies. It's going to look, as I said, like a lawn of bacteria, okay? By reaching quadrant 4, this is when you're going to start seeing all the single colonies, okay? So these are some examples of some really good streak plates right here. Again, see how right here, you guys, this is where we have our first quadrant. See how it's a lawn of bacteria. Okay, see how there's very heavy growth, and you can't even see the single colonies, right? See how it's basically like a lawn of bacteria. And then here's the second, okay, quadrant. Notice how you're starting to, the growth is starting to decrease, and you're starting to kind of see some of the colonies. And then like the third, okay, and then right here you guys can see the single colonies. See how here's one colony right there, 2, 3, 4. Okay, so see right here, this is where we achieved those single colonies that we are really after. Okay, now take a look at this plate right here. Again, see how the first quadrant, it's a pretty heavy growth, okay, of bacteria. The second, we have less, okay. Notice how here, right here, you achieved those single colonies, okay. Sometimes they do only 3 quadrants, by the way. Sometimes we do 4. I mean, the more quadrants, by the way, that you have on a plate, the better, okay? So right here, you can kind of see the single colonies, okay? And then same thing right here. Here's the first, the second, kind of like they got it already. They achieved those single colonies already in the third quadrant. But as I said, better to do 4 quadrants, okay? Now, see how here we achieved those single colonies that we are after? And by looking at these single colonies, can you tell whether this plate is a mixed culture or a pure culture? I mean, how can you tell? Look at the colonies. Do they look the same? Okay. Meaning, do they have the same color? Are they the same size? Do they have the same texture, right? If you've answered no to all of these questions, then what does that tell us? It tells us that this plate is not a pure culture plate, it's a mixed culture plate. And so this is the importance of the streak plates. Do you guys see how right here, when we started off in that first quadrant, can you tell in that first quadrant, just by looking at that first quadrant, can you tell how many different species of bacteria you have? No, right? So in that lawn of bacteria, you can't actually tell how many different species you have. But notice how when you kept diluting it, okay, second quadrant, third quadrant, fourth quadrant, and you kept diluting your sample, and you got those single colonies, it's going to help you figure out how many different species, okay, do you have based on, the look of your colonies, whether they're the same or not. And then, as I said, you can achieve a pure culture. How do you achieve a pure culture from this plate? Well, pick up one of your colonies. So if I want to have a pure culture only of that white colony, okay, then I'm going to go ahead, grab an inoculating loop, pick up a sample, only from this colony. I'm not going to touch any of the other colonies. I'm only going to pick up a sample from this colony. And again, I'm going to do another streak plate. And if I get, if I do that and all I get are the same exact looking white colonies, then I've achieved a pure culture. Okay. And right here, this just kind of explains to you, okay, the steps that we already went ahead and talked about it. So again, read over it one more time and make sure that you guys do know the steps, okay, of the streak plate method. Now there's another term that I want you to know, and that is subculturing. What is subculturing? Subculturing is when you want to transfer some of your bacteria that are already cultured on one medium and you're trying to transfer them over to another medium, okay? So for example, if I have right here some bacteria growing on this plate, remember how I said when you look at this plate, you can see that you have two different looking colonies, meaning you have two different bacteria species growing on this plate. And if you want to obtain a pure culture, remember how I said if you want to obtain a pure culture of that particular bacteria. So if I want to achieve a pure culture of that particular bacteria in that white colony, or if I want to achieve a pure culture of that bacteria from that red colony, what am I going to do? Well, I'm going to grab my inoculating loop, of course, after sterilizing it, right? Never use your inoculating loop without sterilizing it first. So sterilize it first, grab a sample. Okay, so you're going to grab a sample from this white colony right here, and then you're going to transfer it over. Now, up to you though, if you want to transfer it over to a slant, you can do that. Or if you want to transfer it over to a plate, okay? And so when you transfer it over to another medium, what do we call that? We call that subculturing. And why do we, why are we doing that step? Why do I want to subculture that white colony? Because if you get only white colonies on that plate, then great. You have now a pure culture of that, white colony, and you have it now on your slant. Same thing, if I want to achieve a pure culture of that red colony right here, well, I'm going to grab a sample of that one colony. And by the way, only touch one colony. So even though, let's say, here's a red colony, here's a second red colony, here's a third, here's a fourth, should you be grabbing samples from all the red colonies, from all of those red single colonies that you're seeing on your plate? Absolutely not. Just pick one, okay? Sample one of them, and that's it. And then go ahead and transfer it over, or now we know the correct term, subculture it, right, on another plate or on a slant. And once you achieve, only red colonies on that slant or on that new plate, then we can say that you have achieved a pure culture, okay? And then right here, as I said, there are a bunch of videos that you guys can watch to help you out with the streak plate method and, with the subculturing and everything. And there's a virtual lab that you guys are going to also do for this lab, okay? And this is an image right here that you guys are going to use to answer some questions that you have on your worksheet. You guys see that we have here multiple streak plates of pure cultures. So this is a pure culture streak plate of E. coli. Right here is Serratia mersens. Right here Staphylococcus aureus. And then right here Klebsiella pneumonia. So you see, how S. aureus looks like, right? Like how their colonies would look like. You can see how Serratia mersens look like colony-wise. Same thing with E. coli. And then right here, let's say that you have 3 plates right here, okay? Mix A, B, and C. Now take a look at each plate, right? And first thing, figure out whether they are pure or mixed, but I mean, the we like, the hint is right here. They're mixed cultures, right? Meaning that you're going to have multiple bacteria species, right, growing on those plates. Try to figure out which bacteria species do you have on plate A, B, and C. And of course, go back to those plates up there, up at the top, to be able to figure it out.